All right, so a couple of things to talk about today and some changes. Quick reminder, again, night at the library, and I just posted in the AMP website under tutors, they got another tutor. He's going to be there at the night at the library, which is February 21st, 6 to 8:30 p.m. It sounds like they have both walk-in as well as scheduled, and for 227, 228, as well as some of the other biology courses. But I know there were still some people looking for a tutor and they couldn't get in before. But some people may have dropped from the course. There may be some more openings. So do check again if you were looking to get into tutoring and you couldn't before. You should be able to have that opportunity now. Another thing to mention is that we may have a lockdown drill that may occur sometime during this class. If it does, I would ask that, because I can't cover up the windows, we're supposed to keep the doors closed, and we'll mush into the center of the room and towards the back. I hope it may not take much more than 15 minutes. And then what we'll do is continue with class after that, if it occurs during our class time. If not, it may occur sometime after our class, so just be aware. They didn't want to scare anybody after what happened yesterday in Florida. So another thing to talk about. Let's see. I will record the last lecture for this exam, for exam 2, probably on Friday sometime. And then I will post in the AMP website that I got it recorded and posted so that you guys can view it. That gives you the whole weekend to try to go through exam 2 material and study for it. When I come back to class for Tuesday, I will then continue on with exam 3 material, which might have a little bit of review because it's a continuation of bone information. And you guys would then have your exam for Thursday. If you're taking it with us internet students, it would be on Thursday at 1 to 2:15, room 106, Meyer Health Science. And other than that, let's see, what else do I want to tell you about? Any questions about anybody have a problem with me recording that exam so you can study over the weekend? Awesome, good. Okay, another thing. So usually in 227 and 228, we have cat dissection. But over the last few years, there has been a shortage of cats, and occasionally they don't get the order to us quite in time. One semester, it just delayed us by about 2 weeks, and we were able to work around it. But this semester, and the cats weren't just ordered, they were ordered almost a year ago. So they won't get to us until the end of April, which will be a little too late for your guys' class. What that means is we will not have cat dissection in 227. We will end up using more models in the classroom. There is some other dissection things that we will do, like the brain dissection. We still will have that. That's not cat, it's sheep brain. And we will also get to utilize the cadaver more, which is really nice. We haven't always had time to do a lot of cadaver in the classroom. So you guys will get to have more exposure to the cadaver. We won't jump in and do it today or tomorrow with your bone evals, but we will be introducing it more. And we're hoping that we'll get to use the anatomage table if we're able to move it into a room that allows you guys to utilize it more. Basically, it's a ginormous double-screened computer that's laid flat, and you guys can manipulate and a couple of simulated bodies and organs and stuff. So it's kind of nice to be able to utilize that. So if we can get that into a room that we can get into use, then we will be able to incorporate that into the lab as well. So keep in mind, we were not doing cat dissection to learn about the cat. It was a model to learn about human anatomy, because this is human anatomy and physiology. And so it was nice to have that specimen to use, but this way I think you guys will actually be able to focus a little bit more on the actual human anatomy. And your big eval is the muscles. So the other nice thing is that what was I going to say? Lost my train of thought. Cadaver, and darn it, it'll come to me. Oh well. Okay, so human anatomy, and what else did I write down? Oh, I know. I remembered. So you guys were utilizing chapter 12, and you guys noticed in lab news that I had said, focus on just a couple of these areas, not so much this part. And so because we don't have the cats, we can expand that a little bit. Do not stress out if you already fleshed out chapter 12 in your lab composition book. Do not stress out if you already started your cat dissection journal area for, thinking that you would be skinning the cat, because that was the plan up until 2 days ago. And so it's okay. Don't worry about it. You don't need to cross it off or anything else, okay? But with chapter 12, we'll be able to focus a little bit more on origin and insertion that we didn't have time for before with the cat dissection part. So you guys will, when you get into lab this week, your lab instructor will walk you through that. And it's not expected that you knew this change was going to occur. So it's okay. Don't stress. Stress about your bone eval. Don't stress too much. Just get it done. It will be okay. It will be fun, actually. So it will be fun, I promise. So we will focus a little more on origin and insertion. You guys will notice in chapter 12 that there were some tables. And so your lab instructor will inform you and say, focus on this table here. We're not worried too much about the muscles of the face or into the skull, but you will do a little bit more of the neck muscles that we were not including previously. So we used to have them in the past. We cut it out because there wasn't enough time when we were doing cat dissection. We can bring it back now. You guys will see some of these muscles. So we call them the strap muscles. So we're talking about omohyoid, sternohyoid, sternothyroid, thyrohyoid, things like that. So you'll see those in the tables. You can start looking at those. But again, don't worry about trying to go over all those muscles. You will have plenty of lab time to do that. Okay, so bones this week, Thursday and Friday. They'll start talking to you more about chapter 12. And I will end up having to update lab news and make some of the changes. So you won't be going into the cat photo album looking for information because you won't be using the cat. So another thing that I just posted on the A&P website is here's a helpful resource. So of course you'll want to use APR or Anatomy and Physiology Revealed, which is in your Connect site, correct? And then also another good one is Get Body Smart. You do need to use Flash, so if you click on that, it will open into another tab, and you can click on to muscular system, for example, and then you won't have to worry about facial expression or mastication, but maybe you want to look at muscles of the forearm or muscles of the abdomen. And then they'll highlight it for you, external oblique, internal oblique, and your lab instructor will give you hints and tips in lecture, or not lecture, but in the lab part about, external oblique goes like you're putting your hands in your pocket. So internal oblique goes the opposite of that, and they'll talk to you about that kind of stuff. So Get Body Smart is a great resource that you might find is helpful. I just posted it just a few minutes ago for you guys. Any questions about lab? Yes?
I'm actually wondering how you will be simulating the muscle evaluation.
We'll be working on that, and I will let you know. Yeah, we'll be updating and changing a few things so that it's applicable for you guys, because obviously that does change how we were testing you before. So, yeah. So I will keep you updated. I was planning on doing this over the summer break for next semester. I just had to speed up the timetable, right? All righty. So back to our notes. Did I cover everything else? All right. So we'll be covering the same material that we would have covered with cat muscles because the idea was you're learning the muscles of the cat to correlate it with the human, and they were similar enough that that's why we use the cat as a model. And not having them, sometimes students, when they were dissecting, they spent so much time freaking out about the dissection part of it that they weren't always learning their muscles. So when it came to the muscle eval, they didn't even know, like you'd say, well, where's pectoralis major? And they're like, I don't know. Where do you think it might be? Stop thinking about the cat and think, where do you think it is on you? Some of these are very obvious, but you guys freak out and you go like, oh, but it's the cat. So this will actually be, I think, a good thing for you guys. And of course, the whole idea is to learn, explore, and investigate human anatomy. So I think I covered everything. And moving on, where were we? Did we talk about the epidermis? We talked about how the keratin was kind of suffocating the cells. You're further away from your nutrient base, correct? Where's the nutrients going to be from? In the dermal layer, the dermis, okay? So epidermis doesn't have blood vessels. And we didn't quite have time to get into melanocytes. So we're going to start right about there. And so we're continuing with the integumentary system. So melanocytes synthesize pigment, and the pigment or the protein is called melanin, okay? And the cool thing about melanin is that it's, what do I want to tell you first? So let's put it's branching. Site means it's a cell, so this is a branching cell. It has spider-like processes. That reach out to the keratinocytes. So what kind of cells did we call the cells of the epidermis? So they're stratified squamous, right? But because they're also producing keratin, which will waterproof the outside of our integumentary system here, right? The outside of the integument. More specifically, we can call those guys keratinocytes. So all of these guys right here are keratinocytes, okay? They're producing the keratin more on the younger, healthier cells that are closer to the basal membrane, right? The basilar surface. So these guys here are what are producing more of the keratin. And then the cells are dying as they're getting mushed up to the top and all that keratin is being produced. So these guys are the dead cells at the top right here, correct? And so they're not producing keratin at that point, but they're full of keratin. Does that make sense? Okay. So melanocytes are in the epidermis. They have spider-like processes that reach out to the keratinocytes that are actively producing the keratin. So if I draw here here's my basement membrane. Epidermis is on the top part, dermis is on the bottom part, right? Okay, so if I have this spider-like cell, okay, here's my melanocyte, here's his nucleus, and he's hanging out there, okay? And then if I'm just going to draw the other cells in a different color, just so we'll do green. So here are all of my keratinocytes. The majority of the epidermis is made out of what? Keratinocytes. So yeah, we have melanocytes in here, but we also have tons and tons of keratinocytes. Pretend there's a nucleus and all of those guys. I just don't want to draw all the dots, okay? If you really want all the dots, you can put them in your picture. So here we are. Here's our epidermis. We have one melanocyte there, and they'll be spread out, okay? And we have the sun. If you're in Arizona, it's super hot. And the sun is producing UV rays, okay? So and it ends up getting down to that melanocyte. The melanocyte is going to produce melanin. So I'm going to do that with, we'll just do it with red, okay? So the melanocyte produces a certain amount of melanin, okay? That is normal amount normally. When it gets hit by UV radiation or from the sunlight, it makes it produce more because melanin will help protect like a sunscreen for your skin. So it has a certain amount the melanocyte normally produces. If you are getting exposed to sun, you're going to produce even more, okay? So what do I want to tell you here? So melanocytes produce melanin. They produce a certain amount normally, but if exposed to UV radiation or sunlight, it stimulates them to produce even more to increase your protection. That melanin will move out of the melanocyte and into the interstitial space or the extracellular space. And what would we call that process? If it's in the melanocyte and now it goes outside of the melanocyte to the interstitial space, what do we call that process? Exocytosis. So the melanocyte does exocytosis of the melanin. So now it starts coming out here, right? We're getting out here into the interstitial space in between the cells. Now, if the keratinocytes take that into its cell, what would we call that process? Endocytosis, because it took it into its cell. So now you can draw your little dots into all these little keratinocytes. And the more you get exposed to sunlight, the more melanin you will produce, the more melanin you will put out, the more melanin the keratinocytes will take into their cell through endocytosis. Does that make sense? And then remember, these cells, as you go through mitosis, because we have lots of active mitosis going on, it's going to push the new cell now becomes the second row cells. And then you have another new row here, and then you produce another new row. Second row now becomes third row, fourth row. Remember, we have, maybe up to 20 to 40 rows. And eventually that newbie row ended up becoming, after about 2 weeks, it finally becomes your outside apical surface. So he finally gets up to the top. This guy had tons of melanin in him from whenever you got exposed. Maybe you went out sun tanning or something. You went to the beach throughout all of the summertime. So you produce more and more and more. And that's on that top row. So what do they tell you when you go and you get a tan? They tell you to moisturize a lot if you want to keep your tan, right? Because the more you moisturize, maybe the more you won't slough off those outer cells, right? They might say keep well hydrated. They might say don't scrub really hard. Don't exfoliate, because you would be sloughing off those outer cells that had tons of the melanin in it. The melanin is the pigment. So you might carry on that tan, depending on how much you kept exposing yourself, for quite a while, depending on how many times the cells got exposed, how many times you kept producing more melanin throughout the different layers. So the longer you expose yourself, the more melanin you produce. That makes sense, right? So in the wintertime, you're not exposing yourself so much, potentially, unless you do. So after a while, some of that suntan starts to go away, right? And you're not as tan as you were. You're a little more pasty if you look like me. You become even more pasty. So, all right. So, what do I want to tell you now? I want to tell you, so the keratinocytes need protection. That's why they take in the melanin into the cells. And then I want to tell you, we did that. So those little dots, that melanin, it's really melanin granules is what it is. And that's what's exocytosed. Then the keratinocytes endocytose it. And then I want to tell you that the keratinocytes go through mitosis. The cells move up to the surface. More melanin, deeper the tan. If you stop the UV exposure, it would decrease the melanin exposure. So you won't produce as much melanin then. And so over time, you produce less and less unless you re-expose yourself again, right? And then the keratinocytes with the melanin are sloughed off, and the newer keratinocytes no longer had all of that melanin deposited in it, right? And then let's write down all the things I just said. Go away, draw, go back to black. So we produce, PD is produce melanin, which protects the nucleus from UV radiation. Okay, which is stimulated. So STIM period is stimulated. So stimulated, the melanin, it's stimulated by the UV radiation, which would then increase melanin production. And we said melanin was endocytosed by the keratinocytes, right? Taken in by the keratinocytes. Okay. And I wanted to tell you guys something else. So we have the same number of melanocytes between all the different people in the population, but we have lots of different skin shades, right? And some people tan better than others, and like I don't tan, I just burn. So my sister has the same skin tone as me, but she tans and she doesn't burn like I do. However, she just found out she had basal cell carcinoma because I probably slathered on that sunblock like you wouldn't believe, and she didn't worry about it as much and probably didn't put as much sunblock on, right? An artificial way of protecting your skin cells, okay? So we have the same number of melanocytes, but the color of the melanin is different. So it can range anywhere from like a lighter tannish yellowish color for like people that are more pasty like me, to having very dark, like different shades of brown to very dark brown, right? So that's how you will have the different shades of skin color. Same number of melanocytes, but yet different shades of the melanin produced, and the amounts that it produces is going to be different. Okay, so where you have like freckles, you're producing a little more melanin like right there. That's about it. Okay, versus somebody that has no freckles, they're not producing like little splotches of melanin in certain areas. Okay, and this is all determined by your genetics. Your DNA, remember when we talked about gene expression? So you open up the DNA, you read off the message. It's telling you on your own DNA what color melanin that you would be producing, and then that's what's being produced by that cell. That cell nucleus has its information, and that's what it produces all the time. This is his job. That's all he does. He does other things too, but that's what his main job is to do, is make that melanin, put it out there. It's going to protect you. Cool. You also have melanin for when we get into the hair that decides what color hair you're going to have. It's the same thing. So what happens when people get gray hair? The cells are no longer, the melanocytes there are no longer producing color. Sherry likes to say it's producing like air bubbles. So it's just not putting out the protein melanin for the color. So now you have white hair there, or gray, lack of color, right? So there you go. And it really sucks when you start noticing you're getting older, like, I'm getting a little gray. It's even in my eyebrow. For men, they look distinguished. For women, we look old, right? Darn it. Okay, so that's melanocytes. So dendritic cells, another name for dendritic cells is Langerhans cells. You'll hear that term again in 228. So the same doctor discovered two different things. So dendritic cells, also known as Langerhans cells. Have you guys heard of pancreatic islets? So in your pancreas, you have something, another name for them is islet, oh shoot. I cannot talk today. Let's just move on. You don't need to know it till 228 anyway. Islet of Langerhans. It's in your pancreas. You'll learn it for histology in 228. All right, back to dendritic cells, also called Langerhans cells. Macrophage-like cells that protect by phagocytosis. It's also for immune system activation. So what that means, macrophages basically are cleaning up debris, and so things that shouldn't be there, it's going to phagocytose it, so chew it up, pulls it into itself, recycles the components that it might use, get rid of something if it shouldn't be there. So like activating your immune system, it would take in, chew up the bacteria, and put a little portion of it out on the outside of its cell. We don't get into the details of your immune system until 228, where we go into a lot more depth of how we do that process. But that's how you tell your other cells, this is what the bad guy looks like. That's what it means by activating your immune system. So macrophages endocytose the bad things. They use enzymes to break things down. They wander around the basal area of the epidermis. So remember, these cells aren't always like interconnected with those junctions that we talked about before. So this guy can weave in and out. He tends to stay near the dermis. He is trying to catch things before they get past the epidermis, because what's in your dermal layer? Your blood vessels. It's deeper into your body. You could get a worse infection or problem if it gets to that area of your body. So if this can catch it in the epidermis before it gets deeper, that's a positive, right? So that's what his job is to do. This macrophage, this dendritic cell, is going to wander around looking for things. Think of him like the police officer or the sentinel. He's out there looking for, is anybody going to cause trouble here? So if you got a sliver in your finger, okay, he's going to try and get rid of it or figure out what's going on before it gets to your dermis and causes a bigger infection, right? That's what his job is. Let's see. And if they do find something, they can activate your immune system and keep the infectious agents from getting deeper than the epidermis. So these are like little biological surveillance cells, those dendritic cells, okay? Let's see. We have layers of the epidermis. So the term strata means layers. So if we're talking about thin skin versus thick skin, okay, thin skin is over most of your body, and it only has 4 strata or 4 layers. Thick skin is called thick because it has one extra layer. So it has 5 layers or 5 strata. That's the only difference between thin versus thick. Thin, like it's saying, is through most of the body. So skin over your arms, skin over the back of your hand, your face, your thigh, all of that would be thin skin or 4 layers or 4 strata thick. Thick skin, though, would be on the soles of your feet or the palms of your hand, okay? And if you look at the palms of your hand versus and your soles of your feet, does it look different than the rest of your skin? Do you have hair in that area? Only if you believe in old wives' tale, right? Guys, that was funny. Come on. Okay, no, you don't have hair on the palm of your hands or on the soles of your feet. Okay, you have it on the back of your hands, the back of your feet. So that would be thin skin on the backs, right? Or the dorsum of your foot. Remember, dorsum is actually the top of your foot. Okay, but the sole of your foot, the bottom of it. And what else do we have in that area that's different? Friction ridges. We'll talk about that in a second, friction ridges. So let's go through our different layers. So the strata or different layers, we have stratum basale, stratum spinosum, stratum granulosum. Stratum lucidum is the one that's the extra layer in thick skin that you don't have in thin, okay? And then you have stratum corneum. So thin and thick all have stratum basale, spinosum, granulosum, and corneum. But if it's thick skin, it will have that stratum lucidum layer in between granulosum and corneum. That's all. That's the only difference there. Stratum basale, you guys have already heard it, right? The basement layer. So this would be the apical side or the deeper side? The deeper side. That makes sense. So that's why it's the deepest layer. So cool. We got it. Cells sit on the basement membrane. I really should write these out ahead of time because my handwriting's atrocious today. I'm sorry. Basement membrane. Okay. And let's see. What cells do you think we find right on that basement membrane that we just talked about? Definitely keratinocytes and the melanocytes. For sure, right? The majority of epidermis is keratinocytes. You have melanocytes there, but the main cell is going to be keratinocytes. And it's not that you don't have the dendritic cells, but they're not as many of them. Okay, so it's not like we have tons of dendritic cells wandering around. That's also how sometimes things get past your epidermis. They don't always get caught. You can't have 5 million soldiers waiting along the line there, okay? And let's see, and it makes sense that these are the stem cells. Do you guys know what stem cells are? Maybe. You've probably heard of them before. Technically, a stem cell, in the true sense of it, would be this is a cell that could become any other cell of your body. Okay, it has, remember, all the cells except for your red blood cells have the nucleus with DNA that has all the instructions for everything, but then when those cells specialize, they only do what they're specialized to do. A heart cell is only going to be a heart cell. A skeletal muscle cell will only be a skeletal muscle cell. A chondrocyte is only going to be a chondrocyte, okay? But, and there's lots of research going on with stem cells. If you get an embryonic stem cell, it's the true stem cell that you hear about that can become like anything else. In an adult, we're not an embryo anymore. We do have stem cells, but they're already semi-specialized. They don't like to revert and just become any kind of cell. And that's where the research comes in, where they're trying to make some of our adult cells become more like that, more progenitor or original stem cell, because then we could fix things easier, right? We could inject that where you had a myocardial infarction and say, become a myocardial cell, become a heart cell and fix that dead cell there that died because it couldn't fix it on its own very well. Right? So, but not everybody likes to think about taking stem cells from embryos. Usually it's from when somebody's had either a miscarriage or they will take them from the cord blood, right? From, not the cord blood, but they'll take it from the cord area. But not everybody likes the idea of that. So it just depends. So there's research. People can get stem cell injections. Now you understand why some are going to be more applicable or actually become what you want it to become versus stem cells that got injected and it didn't take and it didn't become like a new meniscus for their knee, right? So, all right, enough about stem cells. So these are a single row of stem cells. That's why they're supramitotically active also and can become more and more keratinocytes that move up as we make more rows of it. The stratum spinosum, no, where are we at? Single row stem cells, keratinocytes, mostly cuboidal. Oh, certain cancers start here on stratum basale, okay? Because if that UV radiation damaged the cell, right? And they'll say basal cell carcinoma, stratum basale, stratum basal. This is where it got damaged. And then the cell didn't turn off, didn't have its normal telophase or its normal cycle, end cycle, right? And go through apoptosis. I think that was the word I was talking about earlier. Apoptosis, right? So, and then it's growing uncontrollably and making too much. More and more cells, more and more cells. That's an uncontrolled cell growth. That is a tumor. That is cancer. So if their melanin couldn't protect their keratinocytes and then it got damaged on the stem cell layer, that's where you get that. So this is where most cancers are going to start here. Okay. So, and the reason why they're susceptible to that is because if they go through mitosis frequently, they need to be protected. You're opening up that DNA all the time, right? To make another cell. You're replicating. And you get that UV radiation is radiation. It's still radiation, right? So, all right. Stratum spinosum. So this is several layers thick. And there is a little bit of mitosis going on by this layer, but not much. So if you're going to say where mitosis occurs, what layer would you say? Stratum basale, okay? So I'll put some mitosis, but I'm going to put it in parentheses because not too much. And definitely after this layer, not at all, okay? And it has a spiny appearance because, do you guys remember, we barely briefly talked about it before when we were talking about things. So it has a prickly look. That's the spiny term. Prickly look from the fixation process when they put the preservative and the staining on to make the histology slide. So fixation process. And do you remember those cell junctions? Okay, so when we talked about the spiny appearance, do you guys remember those desmosomes are like shoelaces? So if you fix this area and then you could either peel that layer up, you would see all the little spikies of where it was making that cell junction to the next layer. That's what it's talking about for the spiny appearance, okay? So, and when you're making a cell slide also, you end up desiccating or sucking out the water from the cell and the membrane ends up pulling away, leaving also that spiny star-like appearance. That's the other reason it's called spinosum. And so keratinocytes and dendritic cells are still in this layer, okay? They're still producing keratin at this point, and the dendritic cells are still doing a little surveillance in through that area also. Once we get past stratum spinosum, that's why it says cells are starting to die past this layer, okay? So after stratum spinosum, they're not really alive cells so much. They start dying off because they're getting choked off. They're getting too far away from their blood supply, their nutrient supply, okay? And if we go on to the next layer, stratum granulosum, so now our cells are getting flatter, okay? And it's only about 3 to 5 layers of cells thick. And they're flatter cells. So they're starting to look more squamous or squamous. Okay. They're called granulosum because of the granulars. It has a granular cytoplasm. Granulars are like little specks of stuff in there that you're seeing. Okay. And so you can see the granules in the cytoplasm of those cells at that point. And again, you still have keratinocytes and dendritic cells there, but keratinization is beginning here. So what we say keratinization, we mean that the keratin is now visible under the microscope. So you were producing keratin there, right? But it wasn't thick enough, the amount of it wasn't enough that you didn't see it. But now in the microscope, you're actually starting to see it. So that's why we say it starts there. And the keratinization is where it's actually becoming enough to start that waterproofing at this layer of the epidermis, okay? So keratin in enough quantity to form granules and provide the waterproofing, right? Okay. So stratum lucidum, what did I say was special about stratum lucidum? This is only in the thick skin. This is that extra layer that makes it 5 layers total, 5 strata. So this is a clear layer, lucidum, like lucid, it's clear. You can, they kind of say it's kind of glassy so clear layer. Again, it's only a few rows thick, so not too much. It's about 2 to 3 layers or rows of flat, indistinct cells. So flat, clear cells. And let's see. And then the last one, so, oh, again, stratum lucidum, this is thick skin only. It's another protection against UV radiation. So this area is usually very effective. And skin cancer doesn't usually occur in this layer. But in stratum basal layer is where it could occur if it's going to occur. Stratum corneum is the outermost layer of both, whether it's thin or thick, right? So this is your outermost layer. And about 20 to 30 layers thick. So it can be thicker than this depending on the area of your body. Okay, and these are dead cells full of keratin only. Okay. Dead cells full of keratin or waterproofing only. Okay. So for example, the skin of your eyelids versus the skin of your back, right? Definitely a different layer of thickness. They're both thin skin, your back and your eyelid, but this is way thinner, right? So this would have less layers between all of those layers there. It might be closer to the 20 layers thick or 15 maybe or something like that, right? And it takes our cells about 2 weeks to go from the basal layer to the corneum layer. Okay, and then it takes another 2 weeks to shed or slough that off. So that's why I said earlier that it takes about a month to have a completely new epidermis, essentially. Okay. What do you guys think about a callus? Like you got a callus on your finger from writing? Yeah, maybe. Some people get calluses like on parts of their feet. So what is a callus from? Friction, rubbing, pressure in that area repetitively over and over and over and over. It's not just like one time or a few times. This is again and again and again. And so what it is your body is sensing at some point that you're getting all this extra pressure against like your writing finger. If you take a look at it, mine's on my 4th finger. Most people it's on their 3rd. But if you take a look at your little callus there, and some might have a thicker callus than others, and some may not be too thick if they don't press really hard with their pen or pencils. But if you look at it, what happened is because of that pressure, stratum basale said, oh, do you need a little help? I will become even more mitotically active. I'm going to push more cells up there to help protect you a little bit. So in that one little region, you start going more mitosis, and it's building up the stratum corneum layer. So it's the outer layer. You're just producing more because stratum basale doesn't really know what's going on out there other than you're getting more pressure and friction there. I think I need to help you out. And that's why it's not like the whole hand gets a callus because of one little region that needed a little additional help, right? All right, very good, very good. And what else do I want to tell you on that? If you take a look at this picture, the histology slide or the cartoon drawing, is this thin or thick skin? And why is it thick? Because you have stratum lucidum there. Okay. So again, all you're looking at here is you have your basement membrane here. Okay. Here, this little blue guy, they're saying is a tactile cell, but he looked kind of star-like, so I almost would have called him a melanocyte, but they said this guy's your melanocyte. So he's kind of star-like, and he's going to produce the melanin. He's right on the basement membrane. All of these guys are your keratinocytes. Notice they're getting thinner and thinner, right? So your first layer down here was stratum basale, then you had spinosum, then granulosum was just this area here. Lucidum, because it's thick skin, was just that area. Can you see it's not so easy to see on the actual histology slide? It can be a little tricky. Sometimes it's easier to see stratum lucidum, but it takes a little bit of looking at a bunch of slides to get that one under your belt. And then stratum corneum, look how very squamous looking by the time you get there, right? Basal looks more cuboidal, and it gets flat and thin by the time you're in corneum. And if you look under the microscope, you'll see like, you'll have like a little bit hanging off there and, a little tear there, and it's dead. It's sloughing off. It doesn't hurt there, unless you have like that hangnail that's pulling on the other sensitive things. That doesn't feel good. Now, under the epidermis is what? The dermis. So this area here is your dermis. And what was under the dermis, did we say? Subcutaneous, also called hypodermis, right? Where you also have subcutaneous fat. And where if you had to get an injection, they usually do it sub-Q, hypodermic needle under the dermis. Does that make sense now? Where they get those names? Yeah. All right, so dermis, strong yet flexible skin layer. It is below the epidermis. It has a gel-like matrix. And with collagen, see what the line means with? Collagen. It's elastic and also reticular fibers. Okay. We already said it's highly vascularized, so this is where our blood vessels are that feed the epidermis. And many nerve endings. So will you feel pain here? Absolutely. We also have our dendritic cells floating around here, surveilling things. So that's always good. We also have macrophages and other phagocytic cells too, but we'll just list dendritic cells for now. And this would be the area, dermis, is called your hide. Like when they say they're going to tan your hide. Spank your little bottom, right? So your hide. Or if you are a hunter and you are preserving animal skins, this is the hide area that they're talking about, okay? The dermis has two subdivisions, the papillary and the reticular layer. And I was just barely starting to mention it in the last lecture, where you have this up area here, and it's called a dermal papilla. So this would be a dermal papilla, this would be a dermal papilla, because the darker purple above it is epidermis. This is dermis, this is part of your epidermis. So dermal papilla makes that like this. And this is dermis, this is epidermis. That's a papilla, that's a dermal papilla, okay? The up region of the wave. So papillary layer is made of areolar lucite. There are many nerve endings. So sensitive, really painful when you get the paper cut down to there. Very vascular, because this is what's feeding epidermis. So this is great for passing along nutrients to the epidermal layer because of the blood. If we go to the next layer down, so the papillary layer is thinner than the reticular layer. The reticular layer is made of dense, irregular CT. So the CT is different in these two regions, okay? Dense, irregular is going to give us more of our strength to the skin. So this is why we say it's the bulk of the dermis, because it's thicker than the papillary region. Bulk of dermis is this, is the reticular layer. Thus, it's deep to the papillary layer. There are lots of interlacing collagen fibers. And so both collagen and reticular fibers are made from the same base of collagen protein, but reticular fibers is referring to that lacy network. So it's kind of like this, right? Network of interlacing fibers. So this part here would be the papillary region. This would be the reticular region, where the interlacing collagen fibers are. And then we kind of mentioned before, but here's the actual term for stretch marks, which are stria. So the tearing of that part of the dermis is what causes that look. Okay, so what cell do you think is in this reticular layer making all of these fibers and stuff? Fibroblasts. Blast, actively making stuff, fiber, all your fibrous stuff. If you're not sure what cell and there's, would be making most of your stuff, default to fibroblast. There you go. And depending on what specific fibers or proteins it's putting out will be what that fibroblast is making, right? Collagen versus elastin, that type of thing. So most abundant cell in CT are the fibroblasts, okay? And it's making the ground substance, so that's all the extracellular stuff, yes? And so you have collagen fibers, you have glycoproteins, you have reticular fibers. If something tears, it lays down scar tissue, which means you're putting down more collagen or more dense CT, dense irregular CT. Thus, no blood vessels will be able to weave in through that area. So in this reticular, all this lacy stuff, it means you can get blood vessels through here, right? I should do red and blue for you, right? So we can do blood vessels in here, right? And there's blood vessels up here also. So because this was loose areolar CT in the papillary region, so that definitely means blood vessels can get through there easy. If we're lacy in the reticular region, same thing. You can weave. So if it was regular CT, that's hard to get blood vessels through because it's much more dense, right? Dense regular, you can't really weave your blood vessels through it, but this stuff you can. So if you put scar tissue down and it's dense regular CT, because you're trying to impart strength, you don't want it to tear again, you can't get blood vessels through it. So that's why that stretch mark looks white or lighter in that area, because there's no blood flowing through there, right? And then over time, what happens to a stretch mark? Sometimes it seems like it takes forever. It starts to fade away because you'll start to chew up and get rid of some of that scar tissue and be able to start weaving some of your blood vessels closer into the area again. It may not look like the original tissue, but that's why it starts to lessen as time goes on with stretch marks. All right, skin color. So due to a combo of various pigments. So I kind of jumped ahead when I was talking to you about melanin. Sorry about that. So due to a combo of various pigments, melanin, which we just talked about, type and amount produced are related to your skin color. I was like, where did I write this? So we all have the same number. So same number of melanocytes. We all have the same number, okay? But heredity determines the type of melanin. So the coloration of the melanin, right? So let's see, where did I write that one? I skipped numbers. So people that are more pale have more of a yellow tinted melanin pigment. Darker shades of skin will have a darker melanin that is produced. Also, some melanocytes are more active, thus they'll produce more melanin, and that's the genetically determined side of it. The other way you produce more melanin was what? UV exposure, right? UV radiation exposure. That's how you get your tan, because you got out there some more. So UV radiation and just genetically how much you're going to produce on a normal level is what determines that amount. Also, hemoglobin gives some of our skin color as well. So blood pigment, it's red due to the iron. So oxygen, when it binds to the hemoglobin, looks very red. And it'll be more obvious if somebody is blushing if they have pale skin versus somebody that has darker skin. You might not see as much of the blood underneath because it's not going to be quite as bright because the melanin is kind of covering that up a little bit. And then also carotene. So anybody eat tons of carrots when they were a little kid, or have a little kid that ate tons of carrots and the kid turns orange? That was me. I turned orange. Okay, so also probably if you have a lighter skin tone, it's going to be more obvious that you're an orange carrot head than not. And why do kids eat carrots? Because they're kind of a sweet vegetable. They might enjoy the taste of that versus peas or spinach or something else that you're trying to feed them. And if you're a parent, you're like, oh, good, my kid's eating, I'll feed them more of it. So if they look that orangey to you, it's not detrimental, okay? It's good for their eyesight because in areas where they don't get enough vitamin A in their diet, there's more incidences of blindness. So vitamin A is important. You just sometimes go overboard. So what do you do if you've been eating too much carrots? Just decrease what you've been eating and drink more water, and it will eventually not be so obvious, okay? Going on to skin markings. So nevus is another name for mole. I was going to say with carotene, it's mostly the yellowish pigment is getting deposited in the stratum corneum. Also the hypodermis. Hypodermis is where you have the subcutaneous fat also. So it's vitamin A is also a fat-soluble vitamin. That's why it stays. It doesn't just go out in your urine. So too much vitamin B, some of the B vitamins, when you take it, your urine looks what? Neon after you've taken some supplements sometimes. That's because it was water soluble, it went out in your urine. Not all B vitamins are water soluble, but the ones that are, you'll see it in your urine. But vitamin A, K, and E, they're fat soluble. If you eat too much of it, it gets deposited in your fat cells or also stratum corneum. That's why you'll see it in the skin. So nevus, another name would be mole. So this is just a localized overgrowth of melanocytes. So that's why some people have more moles than others. Localized overgrowth of melanocytes. I'm sorry? Nope, different. A little different. We'll come to that in just a second. So this would be a mole. And so some people have many more moles. They're not the same as a freckle. Freckles are a localized overactive melanocyte. So I have more overactive melanocytes on my body. So localized overactive melanocytes. So not an overgrowth, just an increase in activity is a freckle, okay? And then a hemangioma, these are localized area of blood vessel proliferation. So localized area of blood vessel proliferation. This is not cancerous, but it is, oma means tumor, but this isn't a cancerous tumor, so it's just saying it's a vascular growth. That's why it's a hemangioma. So tumor doesn't always mean cancerous. It means an overgrowth, so it could be cancerous or it could be benign. Benign means non-cancerous. So a fibroma or a lipoma, a lot of people have heard of lipomas, where they'll have like a little, movable growth that they kind of feel under the skin and it's not painful, and usually we don't do anything about it. It's a lipoma. Lipo meaning it's a fatty tissue overgrowth of like a localized area, but I've seen them as large as a softball. So they can be quite large. Somebody else has seen some quite large ones themselves when they've been out in the field. And so, and then the last thing, friction ridges. So birthmark would fall under closer to like the hemangioma part of it. And there are people that'll have like port wine stains and other things. We're not going to go into those details, but just so you know, in your textbook, it gives you a lot of info on that if you want to read more on that part. So friction ridges, these are like your fingerprints, but you also have footprints, not just like when you think like in the sand, you put your foot there and you're like, look, I have a footprint. You have friction ridges on the bottom of your feet also. And so if somebody stepped in a pool of blood at a crime scene with their bare feet and walked around, you could match their footprint to that person by their friction ridges, just like you would a fingerprint. Okay? So don't commit a crime, right? So palm, or at least not barefoot. But they can do shoe prints also, I'm just saying, but that's besides the point. So we have palms, we have soles, prints on your soles. You also have them on your toes. Okay, they help provide additional grip. Even identical twins do not have identical fingerprints. They have similar fingerprints because they were in the same womb environment. They were formed with the same environment. That's why they will have very similar fingerprints. But identical twins, they have identical DNA, but their fingerprints are not identical. Close, but not identical. Kind of a cool thing to know. Now you can like let people know how awesome you are. You know something super cool. Subcutaneous layers, not part of the integument, correct? Because epidermis and dermis were the integument. That's your skin. This is below your skin. Subcutaneous. Cutaneous with skin. This is below skin. So not part of the skin. Another name for subcutaneous might be SQ. You might see that for subcutaneous. Hypodermis is another AKA. Hypo meaning below the dermis. Another term is superficial fascia. Okay. It is designed to pad and protect. It's also an energy reservoir. Why is that? Why do you think it's an energy reservoir? Because all the adipose there, all the adipocytes, right? So you can harvest the fat, break it down, and when you break covalent bonds, you're producing lots of energy. So this is where we can store energy for later use. Some of us store it really well. Can't get rid of it, unfortunately. Thermal insulation also. What does that mean? Thermal temperature, right? So, you know, if you have more subcutaneous adipose, you probably feel more warm, okay? At one point, I lost 50 pounds, and then all of a sudden, I was cold all the time. I lost my thermal regulation there, my insulation, right? So I had a harder time regulating my body. My thermostat in the brain was telling me that I didn't have my insulation anymore. And so then I was cold constantly. I'd never been cold in my life. Now I'm not so cold anymore. I've been packing on a few pounds. I got to work on that again. But anyway, that's why if somebody was overweight and lost a significant amount of weight, all of a sudden it's cold all the time. That's what they're going to describe to you because they lost their insulation. All right, epidermal derivatives. Let's see what we can get through really fast. So these derivatives, meaning appendages of the skin, meaning things made from the skin. So we can talk about the glands. So we have exocrine glands. This can be sudoriferous glands or sebaceous glands. Sudoriferous is talking about sweat. Sebaceous is talking about oil. So if we look at sweat glands, it's saying sudoriferous is another term for it, right? And there are two types. You could either have eccrine or you can have apocrine. So the process by which sweat glands use, we learned already, if you guys go back to the glands that we briefly talked about. Anybody want to take a guess as to how sweat glands move the sweat to the surface? It's merocrine secretion. Merocrine. So when you guys, if you made that little chart that we talked about, trying to, you know, decide like a single duct versus a compound duct and how you would call it, and there were 3 ways you could talk about a gland, and then we talked about merocrine secretion, and now you can add to that and say, oh, this could be an eccrine or an apocrine gland then. So you can add to that chart you were making before. So merocrine gland, and merocrine means that it's doing it by exocytosis. Okay. Are we destroying our apical layer with merocrine secretion? No. This is normal, right? We're not destroying anything. That's a different one. So eccrine is going to be your most common. And you have a high number of eccrine glands in the palms and your soles of your feet. And also, where else might you notice somebody really sweating? Yeah, I guess you could say that. I wasn't looking for that one, but you could probably say that. Let's go with forehead, right? When you see somebody's really nervous and they start sweating, right, across their brow, right? Or maybe their upper lip, you might see them sweat a little bit in here, right? Okay, and this is, again, if you don't remember when we were first talking about glands, pull that chart up and go back and forth with it before exam 2 so you get comfortable with it. So this is a simple, coiled, tubular gland. Simple meant what? One duct, because we're talking about a gland. Tubular was talking about the acinar unit or the shape of it, the tubular shape. That's what the tubular part meant. So the secretory part is actually located in the dermis. Okay, and it empties. So the duct has to go through the epidermis to the surface of the skin. So empties to surface of skin. All right, and the main function of a sweat gland might be what? To cool you off, right? So this has to do with thermoregulation, temperature regulation. When we get too hot. And what it's actually secreting, this gland is secreting, is mainly water. So it's 99% water. Is 99% water with an acidic pH. So it's saying that it's slightly acidic when we're sweating, and what this does is it helps prevent things from growing or living on us, or buggies, things like that. So that's normal, if you want to talk about futuristic movies, books, whatever, right? We are acidic on the outside of our skin, but not so much that we're burning. Like you shake hands with somebody, they're not like melting them off, right? All right, so this helps keep our skin environment less favorable to the other buggies and stuff, okay? So apocrine, the other type of sweat gland, this is mainly going to be in the axillary and pubic area. Where's axillary at? Armpit area, okay? Have you noticed that when you do sweat and you didn't use deodorant or antiperspirant, I guess mainly deodorant, and you don't put that in your groin, please don't, but when you use that stuff, you tend to be less stinky, right? So this sweat is stinkier than the sweat that you just have normally. Not that there may not be a smell to the sweat off of your skin or when you've been running around, but it doesn't smell the same as the BO people talk about when you get sweaty and you smell it from the armpit or the groin area. So apocrine, these ducts empty into a hair follicle. So that's different than the eccrine. Eccrine goes to the surface of the skin. Apocrine empties into a hair follicle. Okay, so that makes sense. Axillary, you have hair unless you're shaving it off. Pubic, you have hair there too unless you're grooming that area as well. But you do have hair and it is a hair follicle. So duct empties into hair follicle. So when you guys looked at histology, you could tell the difference depending on where you saw the gland and where it was exiting to. If you had the right area where it was sliced and you could see it, you could tell which type of gland it was. If you couldn't see that it went to a hair follicle, I would have picked another one to say apocrine. Bless you. All right, so this sweat is like eccrine, but it also has fatty acids, so FAs are fatty acids, and proteins that are decomposed. What does decompose mean to you? Death. Broken down, decomposed by bacteria, which is why it gives it an odor or a smell. You might find that smell pleasant. You might not find that smell pleasant. They do suggest sometimes that the scents that we are giving off can be pleasant to other people. So, for example, they did like some research where they had guys that didn't put any deodorant on, and they had them wear a white t-shirt and kind of exercise and move around a little bit, and then take off the shirts, and then in another room give it to these other ladies to smell and decide whether they liked that scent or not, if they were attracted to that person. So animals use scents to, like pheromones, you've heard that term before. So we think that we have pheromones too, but we just may not be as heightened to thinking about it. Plus we cover ourselves in lots of other smells, like deodorants, antiperspirants, perfumes, colognes, aftershaves. I mean, we scent our bodies with other smells so that we aren't stinky. And maybe that's why people get married to other people and don't like them later. I don't know. Maybe they weren't really attracted to the person, or what they thought was attractive about them wasn't. But have you noticed if somebody puts on a cologne or a perfume that you thought smelled good on somebody else, you put it on yourself and you're like, oh my God. It didn't work with your body chemistry very well, right? So not all scents are the same. They react different with us, with your own hormones and chemicals and pH and stuff that you're giving off on your own body. So just kind of something interesting. So this is thought to be like the sexual scent glands of animals. Have you ever sniffed the blanket or whatever that you wrapped your baby in? Your baby can tell the smell of its mom versus not its mom. So what they did was they had moms put the blanket kind of around where they might rub it near their, you have glands on your breasts also, and when you're producing breast milk and stuff, and then give it to the babies, and the babies liked or didn't like it, and they could tell who the mom was. Pretty cool. And they haven't been desensitized with all the other crap we do to ourselves as we grow up, right? So thought to be like, and not for the baby, it wasn't a sexual scent gland, by the way. No. So sexual scent glands of animals. All right. So really quickly, sebaceous gland is, so we had sweat gland or sudoriferous glands. Now we're going to talk about sebaceous glands. So these are the oil glands. These are simple, branched, yes, alveolar glands that empty into a hair follicle. Okay. It secretes sebum, which is the oily substance. So this isn't watery. I'm really sorry for my atrocious handwriting. I'm trying to hurry, sorry. It's designed to function as a lubricant for hair and skin. It is also bactericidal. So cidal means that it wants to kill the bacteria versus bacteriostatic would be that it's just kind of keeping the status quo. It's not killing it all off, but it's not letting it get worse. Bactericidal, think like suicide, suicidal, so cidal is to kill. So if you see like various cleaning products and you see those kind of names, you'll understand what it's kind of telling you. So this is bactericidal. So kills bacteria. This is a holocrine gland. And what does holocrine mean? This time the cell ruptures. The apical cells rupture. So we'll put cell dies. And overactivity would cause something called seborrhea. So for example, like little babies that have cradle cap, that's what they're talking about. So they're born and you just kind of keep gently washing their head and it eventually comes off. It's not something you don't scratch it off of them or anything like that. It will come off. They might look a little funky. You think they have the mange. It's not the mange. But even adults could get this if they had like a little bit too much oily production and it creates something like a plaque. So it would kind of look like they're flaking off a little bit, but it's kind of stuck to them because it's oily. Also, acne forms when the glands get inflamed, meaning there's an infection. Okay, so acne is due to an infection of that local area. So overactive would be seborrhea. Acne forms when glands inflamed, which means infection, like a local infection of that area. All right, so basically what they were showing you before was if you look at a sebaceous gland, it has a lot of cellularity to it. So this is lots and lots of probably cuboidal cells, okay, tons of them. Versus apocrine with the sweat or merocrine sweat glands, these don't have as much cellularity as these guys did. This is all that, okay? So that's all it was saying there on the cellularity. So what I'm going to do is I will record later, probably on Friday sometime, and I will post on the A&P website. I will continue with the hair and hair follicles and then go on to bones and the function of bones. So when we get to the end of chapter, I think it's the end of chapter, what is that, 7, would be the end of exam 2 material. Okay, so I will record up to that point and stop. I won't go on to exam 3 material on the recording, but I will try to get it in an hour and 15 minutes or less if possible. I do ask questions when I'm speaking to nobody because I want you guys to pause and think sometimes. So no, I'm not talking to somebody. I'm all here. I'm looking up at like nobody, like, what do you guys think about blah, blah, blah. So just somebody asked me one semester, they're like, was somebody there? I'm like, no, I'm talking to myself. But if you have questions over that last lecture that I am recording for you guys in the next day or so, go ahead. You can email me. You can post it. If you post it, I guarantee other people have the same question. Another student may answer it, or myself, or another instructor, and that is fine. But do not hesitate to ask questions. Also, you can ask me questions on Tuesday when we come back, and we'll be moving on to exam 3 material on Tuesday. Or you can stop by my office and ask questions there as well, okay? All right, so let's stop the recording. I'll bring up some quiz questions.